Hi, my name is Jenna, and this is my 3D DNA model. I'll be explaining how I built it, the history of DNA discovery, its structure and function, and why DNA is so important. Part 1. History of the DNA molecule. Let's start with the history of DNA discovery. It took many scientists over time to figure out. 1. Friedrich Michier, 1869. He was the first to discover a substance in the nucleus of cells that we now know as DNA. He called it nuclein. 2. Phoebus Levene, 1919. He figured out that DNA was made of three parts: phosphate, sugar, and a nitrogen base. 3. Erwin Chargaff, 1950. He discovered that DNA has the same amount of AST and C as G. This is called Chargaff's rule. 4. Rosalind Franklin, 1952. She used X-ray diffraction to take a photo of DNA, called Photo 51, showing it had a helix shape. 5. James Watson and Francis Crick, 1953. Using Franklin's photo without her permission, they built the first accurate model of the DNA double helix. Part 2. Significance of DNA. DNA is super important because it's the code for all living things. It carries the instructions that determine our traits. Genetic terms to include dominant and recessive traits. A dominant trait only needs one copy to show up. A recessive trait needs two copies, one from each parent. Homozygous versus heterozygous. If you have two of the same allele, like AA or AA, that's homozygous. If you have one of each, like AA, that's heterozygous. Genotype and phenotype. Genotype is the actual genetic code, like AA. Phenotype is the physical trait you see, like blue eyes or curly hair. Purines and pyrimidines. Purines are A and G. They have two rings. Pyrimidines are C and T. They have one ring. They always pair together, AT and CG. Part 3. Structure and function of DNA. Now, let me walk you through my model and explain how DNA works. Double helix. As you can see, my model is shaped like a twisted ladder. That's the double helix structure discovered by Watson and Crick. Base pairs, rungs. Each rung is a pair of nitrogen bases, adenine green with thymine purple and cytosine red with guanine blue. Hydrogen bonds. AT pairs have two hydrogen bonds, and CG pairs have three. I added those here in the center of each base pair. Backbone, rails. The sides of the ladder are made of sugar and phosphate. I used the green pebbles for deoxyriboose, sugar, and yellow for phosphate. Phosphodiester bonds. These small connectors link the sugar and phosphate units down each side, like a chain holding the backbone together. Questions people might ask. Why do A and T always go together? Because they form exactly two hydrogen bonds, just like they're puzzle pieces that only fit with each other. What does DNA do? It holds the instructions for building proteins, which make up everything in your body, from muscles to enzymes. Can DNA be different in everyone? Yes. The order of base pairs, called a sequence, is what makes your DNA unique. That's your genetic code. Thanks for watching. This is my DNA model, and I hope to help explain how DNA works, how it was discovered, and why it's so important. Have a good day.